Thank you, Dan. Thank you all for coming. If you can't hear me, please just put your hand to your ear or something like that, and I can try and speak up. All right, great. I'm going to talk today, as Dan said, about diversity and functioning in pollinator systems in particular. I'm going to start by giving you an outline of my talk, and I'll give you a brief overview of my research program. I see that there are a couple of people here who are also in my bio talk. My research program hasn't changed in the last 2 days, so that will be a refresher for you. I'll get into some conceptual background about diversity and functioning, and then get into both some field work and some laboratory work that we have done on this topic, and then talk briefly about some future work in this sphere and very briefly reference some broader future work. And then talk a little bit about some of the work that I've done in the realm of environmental policy. So in terms of my research program overview, I study biological communities and the structure of those communities and how this structure impacts their functioning and how it impacts their responses to perturbations. So really the stability of these communities in a very broadest sense. Within this idea of biological community structure, we really focus on two different topics. So first, we focus on the structure of ecological interaction networks and how that impacts both function and response to perturbations. That was the topic of the talk I gave in biology on Monday. The other facet of, I just want to briefly mention, for those of you who might be interested, that work includes some snowmelt acceleration work looking at how climate change might be impacting some of these networks, as well as serving as a way to perturb network structure. So if anyone wants to talk about that afterward, I'm happy to do so. And today I'm going to focus on the other facet of biological community structure, which is biological diversity, and again, how that impacts ecosystem functioning. My lab addresses these questions primarily, but not exclusively, using plant-pollinator interactions as a model system. These interactions are really fun to work on. It's fun to follow bees around in the field and watch them forage. They're really great to sort of engage students, undergrads, in working on these kinds of systems. They're also, I think, very important because of the pollinator declines that are going on around the globe. And given the central role of pollinators in the reproduction of wild plant communities, this is really important for ecosystem functioning in many, many kinds of terrestrial ecosystems, and it also really has direct bearing on our food production system. To address these questions, we use a really wide range of research approaches. These include both manipulative and comparative field studies, lab behavioral experiments, which you'll hear a little bit about today, range of genetic techniques, and we also use a lot of theory and modeling. So one signature of my research program is really trying to tightly integrate empirical and modeling approaches. So we'll jump into some conceptual background here. One of the most striking features of life on Earth is its diversity. And you can see that from this very simplified phylogeny, even though this particular one seems to be extremely vertebrate-centric. But it still gives us the idea that there is quite a lot of diversity in life on Earth. And I think one of the most interesting and important discoveries in ecology in the last maybe 30 years or so is that this diversity is important. It's important for the functioning of ecosystems. This is a picture of the Cedar Creek experiment, ecosystem experiment in Minnesota, run by Dave Tillman, and I think Peter Reich was here last week, is that right? So he may have talked about some of these experiments, and I'll actually show a couple of graphs from one of his papers. But in this experiment and some other experiments like this, scientists have manipulated diversity of plants and really shown that it can have a tremendous impact on the biomass that those communities produce. So the foundational sort of fixation of carbon that runs our terrestrial ecosystems. And it can be important for many other functions as well. Biodiversity can also support ecosystem services, which are these ecosystem functions that relate to human communities. So they can provide us with clean water, provide us with clean air. There's actually even some evidence that having diversity in the community of natural enemies of crop pests can help support agricultural production. So today I'm going to talk about how pollinator diversity can impact plant reproductive functioning. And talk about that in 2 different study systems, both in the field and in the lab. I'm going to start off by talking about the field component of this. And I've got a few slides here that I will try and offset from the rest and these are slides that will have sort of questions or predictions or hypotheses on them with a black background and white text. So the question here in the field system that we ask is, if we lose a single pollinator from a system, what happens to plant reproductive functioning, to plant pollination? And this is an interesting question, in part because theory suggests, we look at the broad theory of diversity and functioning, that we would see no effect of losing a single pollinator species from the ecosystem in terms of the function. So why is that? Here's probably what I think of as the foundational diversity functioning model that Dave Tillman published with others in 1997. And what we see here on the x-axis is species richness. So that's the diversity in the system, and really whenever I use the word diversity today, I'm using that as a shortcut for species richness. On the y-axis, we have plant biomass, so accumulated, again, fixation of carbon in the system. And what we really see is that as species richness increases, this has a positive effect on ecosystem functioning. But that effect saturates out after we hit a number of species. So if we're over on this side, and we lose one species and move just a little bit that way, we won't see any real change, any meaningful change on the y-axis. Okay, and that's also consistent with some experimental evidence. And here's some data from this Reich et al., Peter Reich paper from 2012. So what gives rise to these predictions? So what is the mechanism behind this? And really a lot of this is driven, or thought to be driven, by resource complementarity. And complementarity is really another word for the idea of partitioning of resources among species. So for example, if in this cartoon here, if we have different nutrients that are available at different depths in the soil, and different plant species that grow their roots to different depths, we would get more nutrient availability at the community scale, and that would scale up to actually more biomass at the ecosystem level. So basically, the species complement one another in terms of which resources they use. So that's that idea of complementarity. So if we don't have any overlap among those niches, those resources, what we should find is that biodiversity should linearly increase with species richness. And that's, again, assuming that we would have an almost infinite number of resources and we could just keep adding species and we keep accessing these different resources. But we see a linear trend here. But in nature, what we see is that traits actually overlap, and they overlap substantially among species. And because of that, we see functioning really saturating quite rapidly with diversity. So I'm going to throw out there that these predictions might not actually apply to pollinators. So why would that be? Part of the reason why I think has to do with this concept of phenotypic plasticity, which you probably saw in the title of my talk. And phenotypic plasticity in general is the ability of organisms to change their phenotype. So this is to change their observable set of characteristics. And this can be either through behavior or morphology, and this can be, as well as other dimensions, so biochemical, etc. And that's typically in response to some kind of environmental variation. And that environmental variation can be biotic or abiotic. So a lot of traits in nature are actually fixed. They don't really have much wiggle room for plasticity. One example of that is photosynthesis pathway. Plants can't just decide, hey, tomorrow I'm going to be a C4 plant and today I'm a C3 plant. They have that sort of biomechanical or biochemical rather machinery fixed, and so they can't really change that. A lot of other traits, though, are actually quite plastic. So roots, for example, plants grow their roots and they grow away from often the roots of other plants. Not always, but they're very responsive to their rooting environment. Similarly, if plants get shaded, they'll often grow away from the shade, etc. So this is a morphological response. We also see morphological plasticity in animals. These are two different individuals of the same species of Daphne. The individual on the left was grown in the presence of chemical cues from a fish predator, and it developed this defensive morphology, whereas the individual on the right was grown in the absence of those cues. And certainly, behavioral plasticity is very, very important for animals. It allows them to really express a broad range of different functions by behaving. And certainly, pollinators in particular have very broad plasticity in terms of what they forage on and how they forage. So in these biodiversity and ecosystem functioning models and papers, there's this implicit assumption that differences between species and their traits are fixed. I think this is most clear in models that are done on this topic. So this comes back to that Tillman graph that I showed you before. And if we dig into the paper, we can see that here's this depiction of different species. Each species is one of those white circles. And they have a fixed niche in terms of where they fall in terms of temperature and where they fall in terms of soil pH. And these two axis titles are actually arbitrary, really. It could be any kind of different niche dimensions. So this is very clear that this is assuming these niches are fixed. Again, this is this sort of canonical idea that functions is going to saturate with diversity, particularly when we have overlap among species. Now, first I said, well, maybe plasticity is one reason why we might expect this to be different for pollinators. I think one of the things that comes to mind for many people is that plasticity could actually make this worse, not better. And why is that? Well, think about plasticity as implying changeability, right? So if something is more plastic, it's more able to change, and that means it can be more like other species, right? So it can actually lead to species being maybe more similar, and if two species are sort of so plastic that we can't tell them apart, and they can really cover this broad range of niches, we won't see any relationship at all between diversity and functioning. So it really could flatten that relationship out. And when we think about pollinators, this is a concern. So pollinators really have very, very broad niches. So here we have 3 different cartoon flowers, 3 different pollinators, and all 3 pollinators are able to visit all 3 flowers. This is something that we certainly see in nature. But by contrast, plasticity could also strengthen this relationship between diversity and function. How could it do that? In terms of the pollinator context, we could look at the maybe a very broad range of flowers that a pollinator might visit if it is not really competing with other species. But then in the presence of other species of pollinator, these might really start to segregate out in terms of which flowers they're visiting most. So now we can see that the bumblebee in this case is really focusing more on this purple flower, and each of the pollinator species has picked one plant that they're primarily focusing on. Obviously, this is somewhat oversimplified, but we do see patterns like this in nature, and some of the work I talked about on Monday also was very much in the same kind of theme. So if plasticity is expressed in terms of avoidance of competition, we could start off with plants that might have very similar fundamental niches, but then when we look at their realized niches in the presence of competition, we might see much more niche complementarity and a strengthening then of that diversity functioning relationship. I will say that this assumes that species are really focusing in where they have a competitive advantage, right? That again, the species aren't exactly equivalent, or else we wouldn't see any kind of diversity functioning relationship. Another element for pollinators is that individual level specialization does matter for pollination, but it doesn't in terms of what an individual does for most functions. Now, if different species are different, species level specialization might really matter. But but we're now talking about individuals. So why does it matter for pollination? Plants need pollinators to sequentially visit different individuals of the same plant species so that they can transfer the right kind of pollen between those individuals. So in this case, this bumblebee is really doing a good job of providing good pollen transfer between different individuals of the same plant species. But in this case, this bee is showing very low, what we call floral fidelity. So moving between different individuals of different species. And at this point, it's really not providing any pollination function to any of those species. So there is a little bit of evidence that's out there that indicates that there may be some positive effects of plasticity. This evidence comes from the plant literature, but again, I'm going to try and relate it back to pollinators. So a really interesting paper from Katie Seeding's lab that came out in Ecology that's basically looking at some transplant experiments among alpine plants. There were two species that would, in the absence of a competitor species, but in the presence of conspecifics, each species would uptake only nitrate. Now when they were grown together, one of those species would actually change its biochemical machinery to uptake ammonia instead of nitrate. So again, it looks like we're seeing more complementarity with more plasticity in that case. I'm going to show again this graph from Peter Reich et al. And the line I showed you before was from the second year of that ecosystem experiment. And if we look over time, actually at the 12th year of that experiment, this is what we see. And we actually see this diversity functioning relationship becoming more linear over time. And that's consistent with the idea of plants needing some time to actually express that morphological plasticity and express those differences between the species. So I think some very interesting evidence there. So again, with plasticity, we could see it going either way. We could see it leading to more similarity, more redundancy, which would reduce the diversity functioning relationship, or we could actually see it doing the opposite of actually increasing differences between species, increasing complementarity, and strengthening this relationship. And I hypothesize that we're going to find the one on the right, given those bits of evidence that I just showed you. So this brings me to the sort of central hypothesis here, which is that phenotypic plasticity is going to strengthen diversity functioning relationships. So let's test that. So I'm going to tell you about some field and laboratory work really aimed at looking at that question. Again, in the field system, the question is, what happens when we lose a single pollinator species from these communities in the field? And again, what happens to plant reproductive functioning? Here's that hypothesis again, and that leads to this prediction that we will see detectable functional consequences. So a detectable reduction in plant reproduction when we lose that single species. Again, if we think about this Reich graph, that we'll see something that's more linear, and that means if we move a little bit on the x-axis this way, that we'd actually be able to see a change in the y-axis. But that wouldn't be true for this blue line here if things were really saturating, the relationships were very saturating. And we wanted to do this without having to wait for 12 years. So the idea is we're hopeful that this rapid plasticity in pollinator behavior would lead to this same pattern in contrast to plant morphological plasticity, which might take much longer to express itself. So how do we test this? Well, if we want to know about what happens when we lose a single pollinator from an ecosystem, one way to test that is by doing removal experiments. And that's just what we did. And we did these with bumblebee removal experiments. And these experiments took place in and around the Rocky Mountain Biological Laboratory, which Dan mentioned. And this is outside of Crested Butte, Colorado. We did this with support from the National Science Foundation. It's a really fantastic place to have worked. Happy to talk to anyone about that afterward. And the data I'm going to show in the experiments really took place using an amazing team of undergraduates and grad students, and a lot of this work was co-led by Heather Briggs, who's now a postdoc at UCLA. In terms of these removal experiments, we removed the most abundant bumblebee species in each site. So we worked in 20 by 20 meter plots. And because the identity of the most abundant species changed over space, we were able to do removal for 6 of the 11 species in this broader system. So the data I'm going to show are not then depicting essentially species-specific effects of just one species. We did this for a bunch of different species. And how do we remove them? We kind of did it the old-fashioned way. We learned how to identify these bumblebees on the wing and then just remove them using aerial netting. We worked in each site, and we would compare each site in a control state and then a species removal manipulation state with just 1 day in between so that we could keep the plant communities in that comparison constant between the two, or roughly constant, as close to constant as we possibly can. And another element of these experiments is that we actually kept these bumblebees alive. We didn't do destructive removal manipulations. We kept them alive in vials, kept them in a cooler, and then let them go at the end of the day. And once we figured out how to do this, we had less than 1% mortality of bumblebees. So one question, or one caveat about this experiment, hopefully some of you are thinking about right now, is that this doesn't just manipulate species richness, right? I'm also manipulating abundance of bumblebees by removing individuals from a plot, right? So this is an important caveat. And if we're reducing abundance, that could have a really strong impact on the function. We were very careful in looking at this statistically, but in some ways we got lucky. But we didn't have any barriers around our plots, and as they say, nature abhors a vacuum. So other species of bumblebee would come in, and about a third of the time we actually had greater abundance of bumblebees in our manipulated state compared to our control state. So we were actually able then to do a really strong statistical contrast. And what we found is that actually we didn't see important effects of abundance for any of our outcome variables. We also, this is ultimately about plant reproduction, and we worked with this plant species, the purple flowers here that you see, which is Delphinium barbii. It's a beautiful larkspur. I know you have a similar species in the Cascades. It's closely related. This plant makes a really nice test case for these experiments because it's really gets almost all of its visits from bumblebees. So we're able to sort of take other potential pollinator species out of the equation here in terms of their importance. And in addition, all of the bumblebee species in the system visit Delphinium barbii. And so basically, if we take one species out, we know that there are other bumblebees that can provide backup for that one plant. So in some ways, it provides a somewhat conservative test case. So we would, and I'll say we pre-bagged floral buds of Delphinium, and then when they were ready, when we had several that were in bloom and sort of receptive, we would do experiments in that site. We would remove the bags on the day of the experiment, and then observe bees foraging on them, again in both the control and then separately in the manipulated state. And we would basically trace the entire process here, from bee foraging, capture bees and look at the pollen that they were carrying, look at the stigmas of the flowers to see pollen deposition, and then track that through to seed production. So what do we find? Again, our prediction is that we would see detectable functional consequences of this species. And again, it's underscored that generally theory would suggest that we would see an effect here. So I'll jump right into some results. I'm going to show a series of graphs here that have the control state on the left and the species removal manipulation state on the right of the y-axis. This is on floral fidelity, and the y-axis is the proportion of bees that only visited a single plant species. So we see a significant reduction in fidelity with our removal manipulations. So we're seeing more bee species visiting multiple plant species in a single foraging bout. When we look at the pollen that those bees are carrying, we see again a significant reduction in conspecific or single species pollen carriage. So this is again the proportion of bees carrying only a single species in their pollen load. When we look at stigmatic pollen deposition, we see again a very similar trend where in the manipulated state we found more stigmas that have multiple different species of pollen on them, as opposed to Delphinium pollen only. And finally, when we look at seed set, we see a substantial reduction in fecundity in these plants, moving from the control to the manipulation. So our field prediction bears out. We do see these detectable consequences of losing a single species for plant pollination. And this is work that we published in advance. We followed that work up actually with some work doing the exact same manipulations, but then looking at how species removal manipulations impacted the entire pollination networks in these systems, and found that those removal manipulations actually substantially changed the architecture of those networks. So again, I mentioned that we've done work in 2 different study systems, in the field and in the lab. So I'm going to talk about the lab study, and the first question is sort of, why would we do a lab study? And the idea is that we can kind of test a worst case scenario for diversity and functioning. So if the sort of niches or traits of different species are exactly the same, and if they're fixed, there really isn't any possibility for diversity functioning to relationships to occur. So what does that look like? Again, if all the species are the same, we don't expect any relationship between function and species richness. Again, even if we saw basically the exact same thing in terms of fundamental niche, if we had competition shaping the realized niche through plasticity, we may actually be able to then see sort of a rescue of that functional equivalence and actually see a diversity functioning relationship. So when I was setting up the field experiment, I said that there's this, caveat or condition that the species need to focus where they have a competitive advantage, and that's where we're actually relaxing this condition by working in the lab. And we'll talk about how we do that. So really, the question here is: can plasticity drive complementarity even in the absence of niche differences between species? So doing this using lab experiments where we have set these up so that the bees actually have functionally equivalent niches. This is work driven by my grad student, Carolyn Ayers, and we are using a system that we developed in my lab with computer-controlled artificial flowers. These are built, each flower is built around a radio frequency ID reader. The bees, you can see, are carrying little backpacks, little radio frequency identification tags. So when they crawl into one of these flowers, that reader reads their tag and then gives them a nectar reward. And here's a bit of picture for scale in terms of what this apparatus looks like in our lab. And again, the artificial flowers are built around RFID readers, and we have 4 different types that vary in the rewards that they give bees, so varying sucrose molarity. They vary in the morphology of these petals of 3D printed flowers, and they also vary in scent. So again, these 4 different flower types, and here's how we run the reward delivery system. So we almost basically turned into an electrical engineering lab for a semester to get this whole thing set up. For those aficionados of this kind of thing in the audience, we're using an Arduino system, and basically gravity-fed nectar driven by solenoid valves that are opening for very precise amounts of time to deliver a very consistent nectar reward on the flowers. We used 4 different bee species in these experiments. These are the 4 bee species that we can actually order by mail or easily acquire the 4 commonly managed bee species in the United States. And in terms of this functional equivalence, these bee species are different, right? They're different in size, they're different in all kinds of other traits, and with real flowers, those differences would matter. But in terms of these fake flowers, if you are wearing that RFID tag and you go into the flower, you get fed. It's basically like if you're wearing a backpack and you go through the store, someone hands you a hamburger. It doesn't matter how tall you are, how color your hair is, how long your hair is. Your traits don't matter. If you walk through the door, you get a hamburger. It's basically the same kind of idea. And so this is this idea of functional equivalence. All of these species have the same RFID tags, but if they go into a flower, they get a reward. So in terms of the lab study, we kept bee densities constant, which is nice that we can do that in contrast to the field. We varied species richness from one species to all four. And in the middle, we used all the possible combinations of 2 and 3 species. So there are some caveats here. One of the biggest ones is because we're using artificial flowers, we can't directly measure pollination functioning. We can really only measure analogs or correlates of function that are behavioral. Again, this is also a very contrived environment, so that is an important caveat as well. In terms of these correlates of function based on behavior, we looked at 3. So one is specialization, so to what degree is each species specializing on one flower type. We also looked at complementarity, which is very similar to this idea of specialization, but where it's different is that is each bee species focusing in on a different type of flower. So that's what complementarity is getting at. And then another really important one is evenness. So what's the evenness of visitation across the different flower types? So is the least rewarding flower getting almost as many or as many visits as the most rewarding flower? Again, our hypothesis here is that plasticity is going to strengthen that diversity functioning relationship, and that in this lab context, functional correlates will increase with species richness. So what did we find? So across all 3 of these outcomes, we see significant increases as we increase species richness from 1 to 4. I'm just showing the graph here for evenness, but we also find this trend for complementarity and for specialization. So again, our prediction here that function will increase with species richness is borne out from these experiments. So future work, I'm going to focus on plasticity for right now, and then I'll just blow this out a little bit more to some broader general ideas. So one question is, are these effects of plasticity more general? Generalizable beyond, say, plant pollinator systems? I think the gold standard for doing this is to experimentally manipulate plasticity. And ideally, we would just completely take away an organism's ability to respond plastically to its environment. We then compare it to organisms that were actually, that did actually have that ability to respond, and then test for differences in diversity functioning effects between those 2. It's almost impossible to do this with animals, or at least certainly close to intractable. If we remove an organism's ability to have behavior, that's really going to mess them up in a lot of other ways. With plants, however, there is a potential to disrupt morphological plasticity. I'll get into how we might do that in just a second here. And in addition, this really might make this work sort of more generalizable. So how would we manipulate plasticity? So plants use environmental cues to detect competition from neighbors. And the idea is that if we disrupt those cues, we might be able to reduce the expression of plasticity. One way to do that is looking at the cues that they use for light, which is the red to violet ratio of light. And we can also look at soil, where they detect root competition through the presence of root exudates. So we can disrupt their ability to detect differences in red to far red ratio by using tuned LEDs. And I'll say that basically the way this works is that leaves are green, and because they're green, they absorb a lot of red light. And so the light that comes through after having been shaded is enriched in a far red signal. So we can actually tune LEDs that have a higher red to far red ratio, and then they're less able to detect that they're being shaded. We can do this with LEDs that actually keep the total amount of photosynthetically active radiation constant between these treatments, which is pretty cool. And this is all hypothetical. I haven't done any of this, but this is just some cool ideas. What I think are cool. With the soil, we can actually add activated carbon to the soil, and that binds root exudates. This is something that people have been doing in experiments for decades, actually been over 50 years of experiments with this idea, and it really interferes with the plant's ability to sense nearby roots. So we can actually do this kind of get fairly close to this gold standard idea. At the same time, these manipulations are definitely imperfect, so plants have other ways that they can sense competition as well. So I think we get conservative estimates. And over the long horizon, one way we can maybe get around some of that is by doing some genetic manipulations, things like CRISPR, or doing experiments, very contained experiments in the greenhouse. But again, this would be over a very long time scale. One other, I think, very productive route on this would be to do some modeling to really understand the conditions under which plasticity might support diversity functioning relationships as opposed to make them weaker. I think this work would really be a strong potential funding candidate for the Rules of Life program, which is really focused on genotype and phenotype, and basically what are the consequences for different phenotypic expression. So I showed you this slide of my research program in general, and I think this again is just one small part of work that I want to do in the future, looking at the assembly, at the dynamics, and the scaling of biological community structure in terms of both diversity and in terms of ecological network structure, and again, how both of those impact function and response to perturbations. I'm running out of time here, but I'm really going to just briefly mention that I've also done a lot of policy work, and two of the areas where I would focus most are pollinator declines and management, and on the Endangered Species Act. In terms of this first part, I was part of this very first assessment from the Intergovernmental Platform on Biodiversity and Ecosystem Services, IPBES, which was their pollinator report, and specifically I was on the policy chapter of that, so I was one of the 75 authors of this from around the world. The people, my collaborators on the policy chapter, we also published a science policy forum as part of that, basically saying here are our recommendations, even though we weren't able to make concrete recommendations in the report itself. I've also done a bunch of work on data-driven, taking a data-driven approach to understanding how we should administer the Endangered Species Act, and doing that with Eric Bieber, who's a law professor at Berkeley. One of the things we've done is sort of highlighting some statistical errors in subspecies assignments that the Fish and Wildlife Service has made. And we've also tackled this question of should anyone be able to propose an endangered species for a listing. Based on Fish and Wildlife Services and data, the answer to this is yes. And I'm happy to talk to folks more about that as well. And I wanted to just say a last word, which is that this talk has really been focused on this idea of diversity and functioning. And I think this idea that having greater diversity can lead to better functional outcomes applies not just to ecosystems, but also to the scientific and academic enterprise. And so this is something that I've really worked hard in my own lab group and in the graduate program that I'm now directing to really try to work to increase diversity and representation and equity. Here's just some pictures of folks from my own lab that I've worked with. And again, this is something that is really critically important to me along many different sort of axes of diversity and variation. And I guess a key theme in all of this is really just reducing barriers to people from different kinds of backgrounds to allow them to succeed in science. And with that, I'd like to thank all the folks who helped make this work possible, collaborators and students and field assistants, and also like to thank many different funders who've made the work possible, and in particular for this work, the National Science Foundation. All right, with that, thank you for your attention. I'm happy to take any questions. Questions?
I was curious about the experiment. You said that it was sort of worst case scenario. So you're removing bees, and then you kind of look to see what happens with the plant. And what I was thinking is that, well, what about the bee? The bees that are there, are they in fact more successful? Yeah, they're not going to the similar species. They're not going like to the delphinium.
Yeah.
But is that their resilience? So it's sort of like, I'm trying to think about resilience. So maybe the plant's not doing as well right now, but what if you have like climate change and something sort of happens? So are the bees in fact being resilient because they're able to go to different plants, or are the other plants not maybe as nutritious? Or
yeah, I think actually probably the other bees and the remaining bees in the system actually benefit from the removals. My guess is if we were able to carefully quantify their sort of nutritional intake, that it would probably increase when they're released from competition. And I think certainly, we're seeing them broaden their foraging niche when this happens, and I think to some extent that's helping to provide some redundancy, but it's just not providing complete redundancy for the loss of that one species. Does that get at your question?
yeah, it's sort of, I'm still sort of wondering if, like, what is the meadow at capacity like? Do you have so many bees there that in fact there's too many? Are there fewer meadows? I know that, like in Washington, we're losing meadows. So is it that you're pushing bees into fewer meadows, and then are you kind of reaching, like you talk about richness, are you reaching a point where all of a sudden there's too many of them?
Yeah.
And so what you're showing is in fact that if you take a few away, that the ecosystem is more functional, resilient?
Yeah, I mean, I guess I think our results will show that if we remove a species, it's at least bad for this one target plant species in terms of reproductive output. In terms of are these things at capacity or overcapacity in terms of bees, I don't think I can really comment on that. I don't think we really have any evidence that would help to kind of support one argument. Yeah, Laura.
Yeah. So in that experiment, you removed the dominant species from each plot.
Yeah.
How do you think your results would differ if you removed a rare species?
Yeah. So when I was doing this, this is the first NSF grant I got, and I really wanted to kind of make sure that it worked. So I did work with, by removing the most abundant species, I think that's where we're most likely to see the biggest signal. And I certainly think if we were to remove the most rare species, there'd be a lot less effect in terms of exploitative competition, right? And so we probably wouldn't see anywhere near this kind of signal. So yeah, I think that is a really important caveat, is that we were removing very abundant species. Yeah, in the back. Yeah.
So, thanks for the talk, super interesting.
Thank you.
I'm wondering, so the setup is species richness and ecosystem functioning. What you showed us was kind of different in a way than I'm used to, in the sense that you have plants and pollinators.
Yep.
You manipulate diversity of the pollinators, right? And what you're showing us is the results of functioning from a single species. In the sense it's planted for the field study.
Oh yeah,
so
right.
Plant
Yep.
reproductive success.
Yep.
And so I guess I'm wondering, how does that scale up? So if I actually think of the functioning of the ecosystem,
Yeah.
how would you relate that back to sort of more traditional way of thinking about, say, system scale productivity or?
Yeah, that's a great point. So, this is one area where there's some inconsistent terminology. So some people refer to functions like this as community functions rather than ecosystem functions, because it's not something where you can capture the entire ecosystem in a single number, right? So that's an excellent point. I showed that picture of the Snowmelt experiments earlier in the talk, and one of the things that we're doing in those experiments Well, I guess I'll first say we tried to do this with more plant species, and just with the field team that I had, we just did not have the bandwidth to do it. So you had to pre-bag these flowers and kind of watch the timing of when they were, developing, and do this across the landscape, and get a lot of people out there to remove bees, and all this stuff. We just couldn't do it. We tried, and we just didn't have the bandwidth. Now that said, with the experiments that we're doing now with these snowmelt manipulations, we are actually looking at multiple plant species. So part of the idea is, can we get a sense of reproductive functioning across the entire community? And not only are we doing that, but we're also actually comparing insect pollinated plants to those that are selfing in the control versus the manipulation. And the idea is that if there's no sort of side effect of that manipulation, the self-pollinated species would have about the same amount of reproduction between the two, whereas if it does impact an insect-mediated pollination, you'd see a drop in plant seed set in the manipulated one. So I think it's a very good point. It's definitely something that we've been thinking about a lot. How can we make a more general statement about community functioning in these kinds of systems? Yeah.
The question about the reasoning on that field experiment.
Yeah.
How do we know that, given the observation, the sort of theoretical prediction is that there is a leveling off of the effect?
Yeah.
There's never, in any of the graphs, I didn't ever see it go to zero effect.
Yeah.
How do we know that what you see isn't the effect that's consistent with the theory? And it would be even more if there was more plasticity effect than you're talking about.
I mean, that's I think a valid point. I mean, when we look at the modeling predictions, I mean, they're basically flat, especially if you look at the amount of error in those error bars. And the other thing is that, so when I first put in the proposal for this, before I had any pilot data, got kicked back from NSF the first time, and they're like, this is way too risky. First of all, what we predict is the saturation of functioning with species richness, but also, in all of these things, there's a lot of variation. So in order to pick up a real effect, this is going to have to be a substantial real effect. So I think that's true. but I guess, we don't, it's true that we don't really have anything in some ways to compare by any means. So that does sort of, I guess, limit the inference in that way. Yeah.
So to follow up on Laura's question.
Yeah.
So you looked the dominant bee for each of the plots.
Yeah.
Plots. And then, did you take any measurements of like the pollen loads on those bees? You had them in jars. Did they show a greater efficiency in this? And did you compare them to the other bees that were part of your diversity counts?
Yeah, so we didn't actually look at the pollen that the bees that we removed were carrying. And again, a big part of that was the bandwidth issue I mentioned before. So we were catching bees, we were capturing bees and removing pollen from them that were left over after we did these manipulations, and doing that was, in the field, was a pain. So we just didn't have the bandwidth to do that. It was something that we thought about, but we just couldn't pull it off in the field. One of the things that we did do, however, is we've looked a little bit at how these different manipulations, and basically different species identities of the species that we removed, how that impacted the functioning. Heather has a paper that she's working on right now, looking at basically traits of these bees. So if we have a much more long-timed bee that we removed as opposed to a shorter-termed bee, how does that change? And that's particularly relevant in the case of Delphinium because it tends to be dominated by longer-termed bees. So the shorter-term bees will occasionally visit it. They'll be able to get a little bit of nectar out of it. You can see, this is the nectar spur, right? So you need a long time to really be able to get in there and actually access that nectar. And so, if we remove a longer-term bee, that seems to have bigger effects because it will switch shorter tongue bees over to foraging on this more, whereas when we remove shorter tongue bees, the long tongue bees that were already there tended to still kind of dominate this resource and still have relatively higher levels of fidelity. But we still saw, pooling across both those types, we still saw these impacts. Does that get at your question? Yeah.
Same question, same thing. So those graphs from Peter Reich's papers.
Yeah.
So, so the longer response, longer time horizon response, you would expect it to be stronger.
Yeah.
So, is there, I don't know if anybody is involved in general in this kind of work to study, so, there are events that happen, right? Bees die off and so on, and so you kind of expect the ecosystem response to be growing over time, right? So I don't know if there's any evidence of that sort. And just a point, for these functions, one thing that's bothering me, there should not be a line between 0 species and 1 species. It's kind of like, it was showing that.
Yeah, I mean, it should be 0.
It should start at 1, right? There's something that.
No, it's true. I mean, and when I plotted this data, I was just using the functions that line up best fit from their data. But yes, you're absolutely right. At 0 species, it's 0. Thank you. So, yeah.
I wonder if you could comment more generally. You talked a little bit about management and pollinator services and your work on policy.
Yeah.
I wonder if you could help connect the theory and experimental and field work that you're doing to the motivations for it or the applications or implications of it for management or policy or action of some kind, conservation actions.
Yeah, I mean, so I think if we go back to especially this paper, working with this assessment report from the IPBES, this is exactly what we're trying to do in this policy responses chapter, is put together, what most of the other chapters we're looking at was, evidence for bee declines, long-term trends, what are the drivers of pollinator decline, what are the economic consequences of that. And then in our chapter, we were really trying to say, okay, well, what can we do about it? So this, and we really tried to distill that in this science policy piece, right? So I think that's one kind of tangible, piece that we've produced that really is trying to sort of guide some potential action. Similarly, I think the stuff that Eric and I have done, it is based on the Endangered Species Act. We don't really have much of a legal framework for the protection of biodiversity in the United States, but we do have the Endangered Species Act. Daniel and I were talking earlier, and in some ways, we can think of the ESA as really being more a species protection law as opposed to biodiversity protection, but it's still, it's what we've got in terms of a legal framework for biodiversity. And so, again, thinking about how diversity impacts functioning, we want to be able to have a legal framework that's effectively protecting species so that we can maintain biodiversity. And in the case of this paper, this is something to maybe just give a little bit more context. There is this provision that anyone, it's often sort of NGOs or scientists, can propose a species to be listed by the Endangered Species Act. And many members of Congress said, hey, this is wasteful, it takes up all this money, and really this is just a bunch of environmental activists trying to block development projects by saying this species should be listed for protection, and then that's going to block that development project. So the members of Congress are saying, can this provision, and that's really what motivated us to do this work, is to look at the Fish and Wildlife Service's own threat level data and then map that back to who listed that species. So the species that were listed by citizen petitions actually had a significantly higher threat level as measured by the Fish and Wildlife Service's own data compared to the species that the Fish and Wildlife Service had listed. So after this came out, there was an editorial in the New York Times about it, and I think this is not really a conversation that we're hearing to the same extent. We don't know, did this paper cause that? We have no idea, but I think it probably at least contributed, right? So I think there is some tangible impact coming out from that, and I think this makes a lot of sense because there are 100,000 plant and animal species in the United States. Fish and Wildlife Service can't have boots on the ground everywhere. They can't have taxonomic expertise in every taxonomic group. It really makes sense that the broader community should be able to propose these things if they are noticing that something, hey, really is in trouble.
What I think is interesting is that you have done a really good experiment to show that one species, the removal of one species, starts affecting the whole ecological networks, the functions and how things go. And I think that with what they just had they published last year, I think 70% of the insects, for instance, like in Germany and Austria, they're looking at those, they're gone.
Yeah.
And so it really starts raising the fact, if you think, oh, take one out, no problem, take two out. But in fact, what you're showing is that there was a measurable change and that really affected the growth of a plant, I think is pretty fascinating because it's powerful, I think.
Thank you. Yeah.
So.
Thanks. Anything else?
Questions?
All right, well, thank you again.